Tēnā koutou katoa, nā mana, nā reo, nā karangatanga maha, tēnā koutou, tēnā tātou katoa. Good evening everyone and welcome. I am Professor Marin Tawhai, Director of the Auckland Bioengineering Institute. On behalf of the ABI and Waipapa Taumatarau, University of Auckland, it's my great pleasure to welcome you to Professor Peng's inaugural professorial lecture. I'd like to extend a particularly warm welcome to Peng's family and friends who have joined us this evening, along with colleagues, collaborators, students and alumni. It's wonderful to see so many people here to celebrate this important milestone. An inaugural professorial lecture is one of the enduring traditions of the university. It provides an opportunity to both recognise the milestone of promotion to professor and to celebrate a colleague whose work has made a sustained and distinguished contribution to their discipline, to the university and to society. It's also an opportunity to reflect on the journey that has brought them here. Peng is very much one of our own, one of ABI's homegrown talents. His journey began at the University of Auckland as an engineering student. In 2007, as a Part 4 student in the Faculty of Engineering, he undertook a project entitled Modelling Electrical Activity of Gastric Smooth Muscle, supervised by Professors Andrew Pullen and Leo Cheng, together with Dr. Travis Austin. It was an ambitious project that combined mathematics, physiology and engineering to better understand the electrical behaviour of the stomach, an area that would go on to define much of his research career. Even at that early stage, it was clear that Peng possessed exceptional talent. His project was awarded the ABI Prize for Best Part 4 Biomedical Engineering Project, recognising both the quality of the work and the promise of the young engineer behind it. So rather than taking that expertise elsewhere, Peng remained at Auckland to undertake his doctoral studies under the supervision of Andrew Pullen and Leo Cheng. Remarkably, by the time he completed his PhD, he had published more than 10 journal papers by my count, I might have that wrong, an early indication of both his productivity and the impact his work would have. Over the years that followed, Peng progressed from doctoral student to internationally recognised academic leader, building his entire academic career here at Waipapa Taumatarau. There's something particularly special about seeing one of our own students grow into a professor whose work is now recognised and respected around the world. Today, Peng is recognised as one of the world's leading researchers in gastrointestinal bioengineering. His research has fundamentally advanced our understanding of the electrical activity of the GI tract and, importantly, translated those discoveries into technologies that are changing the diagnosis and management of patients worldwide. His scientific achievements have been recognised through an extraordinary list of honours, including the Prime Minister's McDermott Emerging Scientist Prize, the inaugural University of Auckland Research Impact Award, a Rutherford Discovery Fellowship and numerous other national and international awards. Another dimension that makes Peng's career particularly distinctive is that his research has not stopped at publication. He's consistently sought to translate discoveries into technologies that improve patient care. He holds patents arising from his work and has played leading roles in commercialising his research. He's a founding director of FlexiMap Limited, a founding Chief Scientific Officer of Alimetry Limited, whose non-invasive gastric mapping technology is now being adopted internationally. And he has contributed to the development of further GI technologies through international commercial partnerships. It's an outstanding example of research moving from mathematical models to engineering innovation to clinical practice. But if research alone defined an academic career, we would only have a part of the picture of Peng. To me, Peng exemplifies what it means to be a university professor. He is quite simply the consummate academic. He pursues excellence not only in research but equally in education. His head of department, Professor Piers Kelly, could not join us tonight, but he noted that Peng teaches some of the largest service courses in the Faculty of Engineering, where students have benefited from his ability to make mathematically challenging concepts accessible, engaging and relevant. His teaching has earned recognition for its quality, reflecting the same care, rigour and generosity that characterise his research. Equally impressive is his commitment to service and leadership. Within the university, he has contributed far beyond his own research program, helping to shape the strategic direction of the ABI and the wider university through numerous leadership roles. He's a generous mentor to students and colleagues alike, and someone who has consistently helped build the collaborative culture that defines ABI. His leadership extends well beyond the university, serving the international research community through editorial and professional society activities. Perhaps what I admire most, however, is not any individual achievement, but the way Peng approaches his work. He combines intellectual ambition with humility, generosity, and genuine collegiality. He celebrates the success of others as enthusiastically as his own, gives generously of his time, and leads by example. Those qualities have earned him enormous respect from colleagues and students alike. Promotion to professor recognises sustained excellence. In Peng's case, it also recognises sustained impact on science, on bioengineering, on education, on innovation, on colleagues and students, and ultimately on the lives of patients whose care has already been improved through the technologies that his research has helped create. Almost 20 years ago, Peng received the ABI Prize for Best Part 4 project. This evening we celebrate the continuation of that journey. It's a journey that reflects exceptional talent and also curiosity, perseverance, leadership, and an unwavering commitment to improving people's lives through bioengineering. It therefore gives me enormous pleasure to invite Professor Peng Du to deliver his inaugural professorial lecture. Please join me in congratulating Peng.
Good, Marin. That was such a succinct and high-quality summary of my work. It feels like whatever I'm going to say is going to bring down the average. Yes. First of all, I'd like to pay respect to the Tangata Whenua, people of this land, for allowing us to share and conduct our work and research. Tina koutou, tina koutou, tina koutou katoa. I mihi ana kina mana whenua, no urumuchi aho, ko Peng tui whanu, ko du ti whanu, ko Peng aho, ko Megan, ko tu hua rangitara, tuku tulu, haku tamariki, some of you already met. Just now, it reminded me of that instance of some North Korean researcher interview on TV, and the kids sort of came in the middle of the interview with the BBC. Ko Valerie, Ratu, ko Dominic, ko Beatrice, aku Tamariki, ka nui timihi, kia koutou, tina koutou, tina koutou, tina koutou katoa. One of the most wonderful things about inaugural lectures at this university, the ones I've attended, is people celebrated their cultural backgrounds. I think Professor Paul Nelson, when he gave his one, even gave a bit of speech in Danish. Well, at least I thought that was Danish, because, what would I know? To acknowledge my Chinese cultural heritage, I would like to quote from a passage by a philosopher from almost 2,000 years ago on the importance of learning. He presents that learning is something that carries on throughout one's life, and what one is shaped by what they learn. So begins the hardest part of today's talk. 君子曰：学不可以己，亲取之于蓝而亲于蓝，冰水为之而寒于水。木直中神，柔以为伦，其取中规，虽有高抱，不复挺者，柔使之然也。故木受神则直，经就力则立。君子博学而日参省乎己，则明之而行无过矣。Well, he used a lot of analogies that could very well be explained by material science today, and I reckon he will cough a bit of grief for assuming that learning is an attribute of a gentleman only, but we give him a bit of slack given he was born over 2,000 years ago. And if we assume that he wrote these things, this passage at the very end of his life, I calculated exactly 2,220 years after him, I was born. There's no direct correlation there, it's just an interesting fact. According to what I could remember, and the photographic evidence here, I enjoyed a wonderful childhood, but sometimes as parents we like to compare what we went through and how our children are experiencing their childhood. And I would say that clearly I was very much content with just the running water hose. Our son Dominic there had a water pistol that had a lithium battery in it that does the automatic pumping and shoot water out in pistols, one of his favorite toys. I don't have a great commentary or insight into this, other than to say that technologies have evolved, but what we enjoy at its elementary level remains the same. And if I was to channel a little bit of my inner Chinese philosopher, I would say that the same thing could be applied to science. And I would say that a lot of what we do in science these days are informed by advances in technologies, which is true. But the fundamentals, the hypothesis-driven research, the equality of data, the discussion, the robust debate, and the reproducibility, these elements will remain the same no matter where technologies carry us. My family, or rather my parents, came from a city in China. I think it's the furthest geographic point in Asia from the sea. So when they decided to migrate, of course they picked a country that was surrounded by sea on the other side of the earth. Sometimes I would tell them, Australia is also surrounded by sea, just, too late for that. But again, I think I enjoyed a pretty wonderful childhood growing up in Auckland. I went to Avondale College. One thing I'd like to point out on this photo, the girl in the front row third from the left, her name is Denisha, and he is Prasad's sister. Yeah, so I went through 5 years of high school with Prasad's sister in my class, not knowing that she had a little brother until I came to uni. And then it's been about 15 years knowing Prasad, not knowing he's actually got two sisters. Right, so yes, that's a close, small community. And one event that I think would awake my scientific curiosity when I was in high school, one day my mom said, hey, come to uni, come to a lecture like this. Of course, it was in the library basement lecture theater. And he said, oh, some guy won some award, and he's coming here to give a talk. So I showed up to the lecture by Professor Alan McDermott, who apparently at the time won a Nobel Prize in chemistry. Something I like to say about this photo, though, this will give anybody at School of Chemistry heart attacks the way he's handling chemicals. But never mind. The point was, I was not interested in his talk when I went in, but I was very much interested in what he was doing, making plastic conducts, as he would say, through a process called doping. And he was a great science communicator. And I remember to this day, he ended his talk by reading a passage from The Story of an African Farm by Olive Schreiner, about the perseverance of scientists forging the path to chase the great white bird of absolute truth. And in fact, that is the same passage I still read to my students on my last lecture to this day. We will come back to Professor McDermott's research because what he started decades ago are still informing the cutting edge of my research. When I came to uni, there was a lot of opportunities. I'm very thankful to be involved in research. But I picked this one because this stood out to me. This is called the EPIC initiative. I can't even remember what it stood for. But the idea here is engineers will go out there and interact with the community. Hi, Beatrice, please have your listening ears on. I can hear you. And we worked with the Auckland Museum in using the 3D scanner at ABI to digitally preserve artifacts. And the artifact I remember most was this hapu presented by one of the iwis to Captain Cook on his voyage to New Zealand. So there in my hand held a history of the country that are now called home. And of course, 3D scanner at the time was this really cool thing, much like AI these days. It's like, oh my goodness, you can digitally scan something. And of course, nowadays you can do a much better job with just your phone. Back in the day, the scanning software was terrible. And so I decided to employ a technique called Delaunay triangulation, taught by, covered in the lecture given by Associate Professor Marion Taffa at the time. And I tried to import this geometry in CM GUI. So I had a lot of fun back in my undergrad days doing research and just applying technologies in various projects. And of course, the so-called real research started with Andrew Pullen, whom I've met early on in my degree. This is always going to be a difficult topic given the tragic passing of Andrew, but today I'd like to celebrate the positive and mention that Andrew was a supervisor that exemplified hard work, and he provided all the resources and gave the space for his students to grow. And I often say, monkey see, monkey do, and students often copy what the supervisors do. And so the question that he posed to me, and we investigated as a team, this is important, this is teamwork, was, what was this pathophysiological mechanism underpinning GI motility disorders? What's wrong with the gut? Is the short phrase for this. And this question didn't appear in a vacuum, because in the adjacent field of cardiac space, whom Andrew and Leo have both spent a lot of time on, of course, electrophysiology of the heart was accepted as a gold standard for diagnosis. But it wasn't the case for the gut. So what about the gut? Where did this thing come from? Well, let's dial back the time for another 100 years. Back to the Spanish, I think, the Nobel Prize laureate Raymond Y. Cajal. So he was a neurologist, and at some point he was observing various tissues, and he observed the gut tissues, and he made a comment about some nerve-like cells at the end of motor neurons in the organs innervated by peripheral nerves, the gut. And later on, these pacemaker cells, or nerve-like cells, ended up being named after him, the interstitial cells of Cajal. They're called the interstitial cells because they reside in the interstitial spaces in between the smooth muscles in the gut. And they act like a network of generators or relay stations to sustain the bioelectrical activation and contractility of the gut. Approximately a decade after Cajal described the pacemaker cells, a physician in the States by the name of Walter Alvarez had a patient. This is an elderly woman who's got some sort of cancer that resulted in most of her abdominal tissue being removed. So he could actually see the contractions, the peristalsis in his gut. Again, in a move that would shock any ethics board today, he tunneled 2 wires through this poor woman's belly. And so he recorded these electrical oscillations there you see on top of the slide. He also noted down, using a pen, the time at which these contractions had gone by the electrode, thereby correlating the bioelectrical activation and contractions of the gut. And this was published in a journal called the American Journal of Medical Association. He had this figure and about a 150-word summary. I looked up yesterday. This journal today has an impact factor of 65. I would love to publish in this journal with a figure like this. Absolutely. In fact, I can draw one up now if they accept it. Please. Later on, jump forward another 40, 50 years, investigators sutured multiple electrodes into the gut. Now we're getting more information. We can see the direction of propagation, these waves traveling from the lower numbers towards the larger numbers down the gut. And I also like to point out that number 9, which was sutured on the intestine, the gut has a very different frequency. So different regions of the gut are doing different things. So clearly, the technological logical reasoning here is to supersize and put as many channels on the gut as possible. So began my 2 decades long love affair with multi-channel mapping. As you can imagine, the increasing in numbers not only demanded an advancing electronics, but also a lot of cable management as well. So you can see just what a mess of wiring that is. And once we have a device, it's not enough. We also needed a model. A model here meant something that's alive. So in a little known fact, maybe shared for the very first time today, the very first animal model that studied by the GI group was not the pig, but on a sheep. Was a leftover sheep by one of the cardiac studies by Nigel Lever, and we applied the insight mapping system on the sheep, didn't get any data, and gave up. And later on, we moved to pigs that had much better data. Of course, did you think I was going to show you some bloody pictures of the pig? Of course not. This is a public lecture. Come on, guys. But we have very talented members of the group who, when in the early days was getting hard, we all baked cakes to motivate us. Emily baked the pig one. This is how we did our actual recording. And I baked the brownie with a bit of frosting and licorice as the electrodes. So I want to acknowledge here that these flexible electrodes that I designed, again, was not something that was born out of a vacuum. It was work and initial prototypes developed by Professor Paul Nelson and Associate Professor now, David Budgett. And they kindly shared with me the design files and walked me through all the basics of how to operate in LTM Designer. So again, I'm very grateful that I had these mentors and received help from colleagues at ABI to give me a boost. So I would not have been where I am today without that initial help. So by studying in the pig, we put these electrodes on the surface, again required surgery. From here, we can reconstruct the activation sequence of this electrical activity in the gut. Right? How does the wave travel from A to B? This gave us a lot more information, the most amount of information out of any research groups in the world. I also want to highlight that these animal studies are quite grueling affairs, and they're always a teamwork effort, right? You can see all the people diligently looking at the screen, the people in scrubs there, operated on the animal, a certain supervisor wearing gloves there doing this, excellent. My promotion to professor is about to happen. What you don't also see, always staying out of the photo, but so critical to everything the GI group does, is the effort by Lindley Nisbet, our fantastic animal technician. Without her, none of the animal studies would happen, and she's always there from start to end of every animal, the prep and the cleanup afterwards. She's not here tonight, but if she was, I would like to tell her thank you very much, Lindley, for your past and continued support. We did studies in animal. We know what the stomach looks like, the activation looks like in the animal, but it's time that we attempted to bring this into the clinics. Again, I was very, very fortunate, and I think this is perhaps by design by Andrew and Leo. When I started, there were 2 other PhD students with medical backgrounds started with me at the same time, so we operated as a team. Dr. John Ikbuji and Professor Greg O'Grady from the Department of Surgery. So they both have lent me and the group a lot of access to clinical settings. And to the clinical and hospitals we did go. And this is where the engineers like to play dress up. If not the first, it was one of the few times Andrew showed up to our studies at the Auckland City Hospital. And I remember the first thing he told me over there, Peng, take a photo of me doing in scrubs. I was like, no, guys, come on. We had 15 minutes at the start of the surgery doing this. I'm not taking a photo of you in front of all these people. He's like, do it. So I did. And I'm really glad because according to the media people that talked to him afterwards, this was one of his favorite photos. It's like, look, I'm on the front line doing surgical related work. And I can see he's also got the pin sort of sandwiched in between his neck there, probably writing everything I've done wrong and how to improve them afterwards. But again, monkey see, monkey do, right? So years later, I was like, no, please take a photo of me when I'm in surgery, pretending to actually doing work. And so we did a lot of baseline studies on healthy, patients with healthy stomachs. They were undergoing surgery for other issues at the Auckland City Hospital. Again, supported not just by my fellow PhD students with medical backgrounds, but also by Professor John Windsor and Ian Bissett from the Department of Surgery. So again, similar to the pig, we could tell from these recordings what's happening to the human stomach. I just want to mention that photo there is a picture of a human, placement of this electrode on a human stomach. And I want to tell you the background, how that photo was taken. So we asked a photographer from the hospital to come in. And something you need to understand about these surgeries is that, the operating table will be the height of this lectern, right? And they have a thing called the sterile field. So everything in front of the scrubs and on the operating table is sterile, and it extends up. So the photographer, to take this photo, the photographer scrubbed in. His camera was sterile behind the film sheet. Then he got a stool and stood on the stool, but he was leaning over. But because it was a bit imbalanced, he went to me, hey, you come here, drag me behind my scrubs, and make sure I don't fall into the patient. So there I was, holding on behind his scrub for dear life, thinking, oh my goodness, if he dropped the camera, that's the end of me. Clearly he didn't. It was the one and only time and one and only photo that we took. He did put a scale there though, so good on him. He was very prepared. So what did we discover? Well, something that you may have noticed from all the studies we've done to date is all of our studies were done in piecemeal. We only captured a region at a time. To help us merge all of these activations together, we developed biophysically based models to reproduce the activation of the stomach in pigs and humans. So the animation there on the left is what the human stomach activation would look like. You start with this red activity near the top, and it becomes these bands of activity going down the bottom. This is what we gathered from our high-resolution mapping studies. And 4 years later to be exact, other independent researchers in Boston General Hospital did MRI studies where they described similar bands of contractions going down the human stomach. So in that way, somewhat indirectly corroborated our evidence. But I would like to say that I will show you later, there's now new evidence enabled by technologies that would challenge this view. Right, so let's talk about one of the most important jobs of a researcher, which is to go out there and tell people about what you do. So I went out there and told people what I do in America. All right, I'm sorry to my American colleagues, but this is my first time in America, San Diego, attending the Digestive Diseases Week. Also my first time in America, by the way. Oh my goodness, where do I start? Lots of flags everywhere. Oh my goodness. Lots of guns, crossbows, hunting shops, okay? Somewhat expected, fine. What I didn't expect is at this conference, they were giving out these really ugly but obvious looking purple bags. So you walk around downtown San Diego with those bags like you're members of a cult. And my supervisors would also, every morning sort of looking for the cheapest place to have breakfast, and here is Andrew and Leo arguing whether they should have a bagel for $10. By the way, the decision was no. Andrew literally said he couldn't justify paying $10 for a bagel. It actually happened, right? Leo can confirm. He was in the photo. And the final thing that happened was there was an explosion. Yes, there was an explosion. A gas pipe exploded near the convention center, and all the local news got in. And Andrew and John there even got on TV. So this was my first This was my first time in America, and I don't remember what I presented, but this I remember to this day. Oh my goodness. And the reason I wanted to talk about America, no political inclination there, the reason I want to talk about America is it is where we did a lot of our studies in the diseased patients. So we did a lot of our recordings in the patients. Now these are patients with gut issues, so we can figure out what's wrong with their gut, at the University of Mississippi Hospital in Jackson, Mississippi. There, we were helped by Professor Tom Abel and Dr. Chris Lahr, the surgeon. They were doing implants into people's stomach, try to cure them, but we had an opportunity, again, 15 minutes or so during each surgery to take some recordings from these patients. I remember on this trip, Tim joined us I think he drove all the way from Michigan, and it was quite a grueling drive. And he often tells people, and I won't steal your thunder when you give your professorial seminar, Tim, it was a grueling process, and you pretty much got into doing human mapping right away before you started your PhD, so that was cool. So what we discovered from all of these efforts was, yes, there's people with sick stomach also have abnormal activities of bioelectrical activation. So on the left, you would have the normal activation going down. On the right, you would have activity that would begin in the center of the stomach. And when this happens over quite a long period of time, you can imagine the efficiency at which the stomach empties its contents would be diminished. And later on, we've also through pathological evidence, microscopic evidence, counted these pacemaker cells in the gut, and yes, discovered in these patients with diminished gastric functions, they also have diminished pacemaker cells. We didn't stop there though. We also employed animal models that had a genetic knockout that changed the structure of these pacemaker cell network and coupled it with our simulations to show that as these pacemaker cells degrade in the gut, they will obviously change the propagation activity. So we had attacked this problem from multiple angles. So what we've done, just to give a very quick summary to date, is we've basically analyzed gut activities at various scales. From very invasive surgical scales, later on moving to less invasive K-hole surgery, minimally invasive endoscopic delivery, and non-invasive delivery. But all of the so-called, as it gets less invasive, obviously it becomes more clinically applicable. But there's trade-off there, obviously. As we get less invasive, sometimes we lose a bit of resolution. But each step of the way is built on the data and evidence presented in the preceding step. I also want to highlight that the recording development doesn't stop there. This is now where I like to bring back, it's called a callback, Alan McDermott's work on conducting polymers. Today, we are printing these conducting polymers using an actuator at the School of Chemistry. Yes, so they have a fantastically flexible substrate, biocompatible, and offers us a way to functionalize these inks as well to record not just bioelectrical activity, but other types of activity is in the gut. This process of printing, in recent years, we've just managed to crack, previously used to take about 4 to 5 hours. But by reconstituting the ink of the conducting polymer to a eutector gel, this now takes about 15 seconds. It's always going to come to this point, so I'm going to keep it brief. I just can't imagine how difficult Andrew's passing was for his family. But it was also obviously very difficult for me. I was at the end of my PhD. Andrew passed away. I'm very, in a way, grateful for Leo for picking up all the pieces and holding it all together to see the group through the transitioning period. I'm forever grateful to Andrew, and again, to this day, every year, I pay a tribute to him, to my students. I will tell him, the way you're suffering my learning method, this is the person you can thank. But there was still a lot of uncertainty, as you can imagine, after a PhD. Now the charismatic PI was gone. Should I have a change in career directions? I don't know, but Leo had a lot of good ideas, and he said, why don't we look into the cellular evidence related to stomach dysfunctions. Now this required linking activities across multiple scales, and luckily for me, I just happened to be a member of an institute that does this thing called the multi-scale modeling, where we look at the activities and use mathematical and biophysically based equations to relate how everything translates from protein level all the way to the whole body level. This was great, and certainly one of the defining signatures by the institute. Over the next few slides, I would just like to give a quick run-through of some of the things that we've been looking at. The first thing is the enteral chromaffin cells, or the touch sensors of the gut. These are the cells residing in the epithelium of the gut. They are sensitive to mechanical stress, and a millisecond of mechanical perturbation could result in tenths of seconds of calcium transient in the gut, and it would inform the downstream serotonin release. So critical to gut functions, and more importantly, this might be a target we can change to keep the gut young. And what we've been doing is looking at a cross-section of the gut, use a bit of biomechanical modeling to predict the areas of the gut that would experience high stress, and matching that to transcriptome data provided by our collaborators at Mayo Clinic. We've also, going back to that original hypothesis by Raymond Y. Cahal as a neurologist, is this a neural thing, or is this some sort of pacemaker cells within the gut thing? Because remember, Cahal was a neurologist, and there are two schools of thinking in the gut field. There's the neural stuff that's driving everything, is these pacemaker muscles that's driving everything. Of course, the truth is probably somewhere in between, but we've been looking at the neural component as well. The reason this is important is because the vagus nerve specifically provides a target to modulate the gut. For example, if you stimulate the vagus, you can actually extend or expand the gut to a much larger volume, while maintaining its contractility. Now, I'm going to present a piece of evidence that will, I wouldn't say upend, but challenge the assumption, one of the data I presented earlier, or even this one, looking at the rings of activity in the gut. And I also like to mention that another reason for looking at the neural stuff is, of course, neurons are often targeted by diseases. Right? One such disease we all know is diabetes. Right? Diabetes kills nerves. Therefore, it may influence gut functions. So this is again through collaborators at Mayo Clinic. So they have obtained or utilizing a genetic variant model called GCAMP model. So this is a type of genetically modified animal model that would make the calcium activity in the interstitial cells of Cahal autofluorescent. It's wonderful, right? You don't need to put any sensors on the stomach, put it on the microscope, you see the activity, calcium activity, but still activity. So, and furthermore, we also induced diabetes in these, some of these mice as well. Now, I hope you can see that it's a bit faint. I probably should turn the lighting to. Oh, this I'm teaching already. Thanks, Carl. It's a bit faint, but these white dots, but they basically represent ICC, or pacemaker activity. This is a diabetic stomach. You can see the type of rotating re-entry waves that happens in the middle of the stomach. So this is, in a way, confirming, yes, when there's nerve damage or when the stomach gets sick, again, there's calcium electrical activity would become dysrhythmic. But what it actually challenges is the assumption that these activity will become a band, because clearly it's a strip, right? It's not a band, it's a strip. What's going on? Well, I don't know. That's the wonderfulness of science, right? But what we do know from this study is that depending on the amount of the stomach that is active, so the smaller region of the stomach that is active, the longer the gastric emptying, GE times, becomes. So again, your stomach has to remain active to pump out all of its contents. Back in Auckland, we've started collaborating with the cardiac energetics group, with Tom Pham, looking at developing these mapping methods. This is now back to electrical stuff in diabetic rats. So we've just finished with Cole Driscoll on the first series of experiments as of yesterday, and he's getting stressed out analyzing data. See, he's here when he should be analyzing data back at his desk. But this is to be seen. So in the final segment, I'd like to just quickly talk about the so what question, right? Marin already kind of highlighted it, the so what question. You know, there is an increased emphasis on producing some sort of outcome beyond publication for research in New Zealand. So let me tell you. This is how we used to record activity from the gut non-invasively. You have a very not happy looking participant there, all wired up, sitting in a chair for about 4 to 5 hours. Lots of wiring, it was a mess. So how do we improve this? Now we know that electrical activation of the gut is important. Well, this is around the time when Allometry Limited was launched. I have to say though, you know, we celebrate Allometry's success, but it was pretty hard going in the early days. So as you can imagine, when things get hard, we bake a cake. Yeah, cake would be a good start to motivate everything. I just want to sort of share with you this background story, right? So what you see there in the photo on the right is a cleaning test. So this is all part of the regulatory approval side. We had to import special dirt, clear biosecurity and everything. So we can smear these dirt onto these devices, and then we have to clean them. I had to clean them. That was me doing it. I had to clean them, right? After I've cleaned them, I have carefully sealed everything in a bag individually and post them to a lab in Japan for analysis. I don't know why. I mean, this is just one of the thousands of tests that we did to get the device up to scratch. And up to scratch we did. We ended up creating an ambulatory, that is something you can wear, device. The blue patch would stick over your abdominal area. The black reader there will clip onto the blue patch, and everything is beamed to the iPad mini via Bluetooth. And afterwards, all the data is processed on the cloud. There's a lot, obviously, people have seen about allometry, but I just want to come down to a specific research question that I've been interested in. This actually came about when ABI celebrated our 20th foundation thing. April, Fiona Lathander from Liggins came to Leo actually and said, well, I'm interested in the Parkinson's disease, and do you think we can actually do some recordings in the gut? And from that conversation, we did a little research collaboration. We recorded from a group of Parkinson's patients. I want to sort of just walk you through this analysis. This is real data. Top is a healthy control. Bottom is a Parkinson's patient. If you look at the top, there's a faint blue line. That's when the subject had a meal. And you see the bright yellow activity. That's on about 2 hours. This is your stomach after receiving a meal, working hard to digest and empty its contents. And afterwards, it goes back to normal. Okay, this is happening in a healthy stomach. In the Parkinson's patient, in the specific case, you can see where the band is lighting up. It's actually much delayed, 2 hours after eating a meal. The reason this is important is because Parkinson's patients often take levodopa, that's the anti-Parkinson's medication, just before a meal, hoping that this medication would literally wash down into the intestine with their meal. So if there's a 2-hour delay, that means there's a 2-hour delay in their body getting the effects of the medication that they needed. So simply knowing this time delay in their stomach would significantly impact the efficacy of drug regimes in Parkinson's patients, because it's not a one-size-fits-all. Everybody have their own regime. It's sort of a trial and error approach at the moment. So hopefully this will give them a bit more objective measure. So finally, of course, there's acknowledgement. All of the founders, colleagues, everywhere, collaborators, students, patients involved in the studies. None of what I do could happen without their support. And I also just want to highlight that this is a note that I made about 15 years ago, highlighting what I understood at the time to be the various pieces informing gut functions. At the time, I think I only sort of focused on the top left quadrant there, electrical, but I'm glad since then, in the 15 years' time, we've managed to answer or at least investigate some of the mechanisms in the other quadrants. There's still much that we don't know about the gut, and I think that's one of the fascinating stories of our time. As academics, our pursuit is not just research. I think, especially now as a parent, I am very much interested in education, not just teaching Tamariki, the stuff, but I think it's more important for us to inspire so that they would have the fortitude and the willingness to pursue a career in research. But I think most importantly as a researcher, and this is not a profound insight, but most importantly as a researcher, we should stay and remain curious. Thank you very much for your attention. Enjoy the evening. I will not be taking any questions, by the way, so just go enjoy the kite, please. Do you want to close, or that's it? Okay, so I've been told to... apparently I've been told I still have to answer questions, but perhaps over drinks and food, please. Thank you.